Good afternoon, everyone. My name is Elisa Sarmiento. I'm a PhD student in the Real Jardín Botánico Madrid. The work I'm going to talk about is the part of my PhD thesis. And there are more authors part of this work that they are not mentioned there, but they are in the book that all of you have. So, all sea turtle species, as we know, are threatened. They face several threats during the life cycle. It is during the sea stage, in migration, mating, and feeding, also during nesting, and during the embryonic development, when they have to face the presence of predators, rain and inundation, and also the presence of pathogens as Fusarium solani, which has been described as causing high mortalities in Boa Vista Island. The picture is shown there. It is how it looks like the nest infected, and it is how it looks, the fungus looks like in the CM microscope. Fusarium is a genus that belongs to the Ascomycetes, and they are worldwide distributed fungi. Fusarium solani inside this genus is a complex of about 50 phylogenetic species, most known as phytopathogenic fungus, some of them opportunistic pathogen for animals, and, as I said before, pathogen on sea turtle eggs. But what we know about these fungi and the incidence or effect of these fungi in sea turtles is based on the work that we have been doing with Caretta caretta in Cape Verde, which is one of the main populations in the world. But what about the rest of the species? Is it present there? Is it causing problems in the rest of the world, in the rest of the species? So there are a few works that name the presence of them in many of these species, but we wanted to get further to know which is the incidence and even the influence of these in these nests. So for this reason, we have two objectives for this work. One was to assess the distribution of Fusarium solani in other sea turtle species and nesting areas, to identify conditions favoring the disease development. For the first objective, we have been collecting samples from 6 of the 7 species and in different nesting areas in the world. The fungi has been isolated from eggshells showing symptoms, as is shown in the pictures, and also for embryos. Following a technique that allows us to grow and obtain pure cultures of these fungi in solid media, in a garden. These fungi, the pure cultures, have been morphological characterized and molecular characterized by the observation of mycelium and spores, and also amplifying the ITS region of the ribosomal DNA. Also, we have run phylogenetic analysis with these ITS sequences of Fusarium solani from sea turtle isolates and also from other animals and plants, also another Fusarium species, to compare in the phylogenetic analysis using the phylogenetic tool PAUP-MIS-arrays. And what we have seen is that they clearly separate a group that is Fusarium oxysporum, another Fusarium species, and one big group which cluster all the Fusarium solani isolates. What is interesting here is that inside the Fusarium solani clade are clustered in subclades A and C, all the sea turtle isolates, from the different locations, being the majority of them clustered in subclade C. So what we can until now is that Fusarium solani is present and may be causing diseases in the Pacific, Atlantic, and Australian coast. For this, we can say Fusarium solani is widespread and present in all sea turtle species tested, And these isolates belong to two groups that are molecularly different from the majority of Fusarium solani isolates so far described, that are clustered in the other clade B that I named before. For the second objective, to identify the conditions favoring the disease development, I ran several physiological analyses using evaluating the growth rate of representative strains in the strains from sea turtles and also from no-sea turtles to compare, and their conditions of temperature from 21 to 37 degrees Celsius. To evaluate the incidence in the natural nest, we have been monitoring a number of nests and documenting the presence of clay, vegetation, and inundation that may be favoring the growth or presence of these fungus in the nests. We have calculated the mortality rate and the incidence of Fusarium solani as the number of dead eggs that present symptoms by this fungus. The results of the physiological analysis show that the Fusarium solani sea turtle isolates are the optimal temperature of growth of these isolates is 27 to 30 degrees Celsius, while the non-sea turtle isolates that are clustered in subclade B has a lower optimal temperature of growth of 27 degrees Celsius. Which interestingly, the 29 and 30 that present the sea turtle isolates coincides with the optimal temperature of incubation. For the evaluation of the condition favoring the disease and the incidence of Fusarium solani, we have seen that nests that present clay, the incidence of Fusarium solani is 78%. It means the 78% of the death of the mortality rate can be attributed to the fungi here, while those nests that present the effect of inundations, the mortality rate attributed to Fusarium solani is 23%. When it's compared with the nest that does not present any of these factors, clay or inundation, which is lower. With this, we can say that clay and inundation is favoring the presence and incidence and even the effect of these fungi in the nests. As conclusions, we can say that the sea turtle isolates have physiological properties that are different from modern Fusarium solani groups, which may represent an adaptation to nest environment, and sun composition and wetness caused by the presence of inundation or rain seems to favor the disease incidence. And as future directions and for my work are to study the influence of identified factors on disease development under laboratory conditions. Also to study some interactions that may be between fungi and bacteria that are present in the nests, and to study the colonization process using different microscopy techniques. Also to study the prevalence of clades and population studies using different molecular markers. With this, I just want to thanks to the Real Haven Botanical, which is my institution, my directors, the directors of my thesis, the International Sea Turtle Society for the grant, and the rest of the institutions and ONG that made it possible this study. Thank you very much for your attention.
Do you think that some of the mortality, do you think that the mortality that you're seeing is being predominantly caused by the fungus, or do you think the fungus is attacking or eggs that would have died, either would have died anyway or have already failed to develop?
Yeah, well, actually, when we try to evaluate the incidents, you see that yes, Fusarium solanum is present in the 100% of the nest. It's present in all the eggs, but we only evaluate those eggs that are dead. For that, we can say it may be Fusarium solani, but we can say it's Fusarium solani, which when it's dead, it presents symptoms, and we see development, obviously, because sometimes you see no development in it. So that's how we evaluate it. Anyway, some data I haven't talked about. We made an experiment infecting the eggs in under laboratory conditions, and that yes, Fusarium solani caused the death of.
We have time for one more question. Okay, good. Thanks very much, Melissa.